Good afternoon, everyone. Today we have the pleasure to have Éric Martens visiting us and giving us a seminar on the human gut microbiota and the problems associated with it. I will briefly introduce the works of Éric Martens. Éric has had his PhD in 2005. He studied between 1999 and 2005 at the University of Wisconsin-Madison. It's in the north of the United States. And his subjects there was to study some nematodes that kill insects, and in particular they have a particular flora, if I understood well, that enables them to kill insects efficiently. Then he went for his postdoc, and he did his postdoc with Jeffrey Gordon in St. Louis at Washington University in St. Louis. And as you all know, Jeffrey Gordon is one of the pioneers of studying the gut microbiota. And this PhD background and his postdoc background, Éric went to the University of Michigan in a small city called Ann Arbor. Michigan is also in the north of the United States, central north, central east. And at the University of Michigan, Éric has now a research group that started in 2009, and he will tell us what he is doing there at the light of what he did with his doctoral studies and his postdoctoral studies. Éric, you're welcome. Welcome to Marseille.
Okay, I'll try and use the microphone. It sounds like it's working pretty well. I'd like to thank Bernie for that introduction and for the hospitality so far that he's shown me and his lab shown me.
Hospitality is also thanks to Michel Rancourt and this organization here at the Faculty of Medicine. So we must thank them.
And thank you as well. It's been a wonderful day yesterday and so far today, just talking about some ideas. To continue a little bit on about Bernie's introduction, my background is really in bacterial genomics and physiology, and as you could have guessed from Bernie's introduction, really a theme towards non-pathogenic beneficial microbe interactions, which is one way, arguably, that bacteria impact our lives more so than the pathogenic ones, but we tend to turn our attention, for obvious reasons, to those that occasionally harm us. The symbiotic aspect that I'm going to talk about today has to do with complex carbohydrate metabolism by many bacteria that live in our distal gut, which is one of the features that they've evolved to help us with in our normal biology. So as microbiologists and as folks interested in medicine, you probably know that we're not alone. We have microbes that exist around us. Many of them exist on our body. It's estimated that we have about 10 to the 14th bacteria just living in our distal GI tract alone. So some people would estimate this to be 10 times more microbial cells than our own cells. And the reason why we need these organisms for digestion is that our digestion is good at digesting things like protein, fat, some forms of starch, and simple sugars like sucrose and lactose. But the vast repertoire of polysaccharide linkages that are out there in the biosphere, that are in plant cell walls, etc., are recalcitrant to digestion by our own enzymes, and therefore we need these gut bacteria to help us do this. So the remaining dietary residue from what we eat, which is rich in polysaccharides, glycans, transits to our distal GI tract, and it feeds that dense community of microorganisms that then help us digest these nutrients. And they do this by fermenting the polysaccharides that enter their habitat to molecules like short-chain fatty acids. These are molecules like acetate, propionate, and butyrate that our bodies, in some cases our colonocytes, have evolved to absorb as their preferential nutrient sources. I still think one of the nicest ways to visualize this effect is old data from Frederick Backhead, when he was in Jeff Gordon's lab, did a very simple experiment where he looked at a group of age and litter-matched germ-free mice. That's GF here. So germ-free mice lack any associated microbes on or in their bodies, so they have no microbiota. And these are 12-week-old germ-free mice. And as a comparison group, we looked at conventional live mice, this CON-D group, that were basically raised germ-free for the first 10 weeks of their life, and then given a fecal transplant for the last 2 weeks. And then he sacrificed them all and compared their body fat percentage. So you can see only having a microbiota for the last 2 weeks of this 12-week lifespan of these mice increased their total body fat by about 50% relative to the germ-free control. And if you reciprocally, if you measure how much food these organisms eat, they reciprocally eat more food in the case where they gain less weight. So just a nice way of showing by the presence or absence of a microbiota the metabolic augmentation, the metabolic enhancement that you can get from having bacteria in your gut. So in terms of who's actually there in our gut, there's about 100 phyla of bacteria and archaea on the planet, and our guts are restrictive to that microbial biodiversity. So about 100 total phyla that are on Earth, we have less than 10 of them in abundance in our gut. And of those 10 that are there, there's really a handful like these 5 up here, gram-positive firmicutes, the gram-negative Bacteroidetes, actinobacteria, proteobacteria, and so on, that are abundant. And typically these top 2, the firmicutes and the Bacteroidetes, are the most abundant. Both of these groups of organisms, as well as some of these others, are good at degrading complex carbohydrates. Some of the early studies done in the 60s and 70s implicated that the Bacteroidetes have particularly broad catalytic abilities towards complex carbohydrates, so they may be very important in driving this metabolic phenomenon. That old idea is shown here in a newer reference from this university, from the Hendershot lab. And what Bernie and colleagues were doing here was looking at the distribution of carbohydrate-active enzymes. So these are the enzymes in these organisms that help depolymerize those polysaccharide substrates and give us that nutritional benefit. And what they're showing here is the scale of these circles at the end of these branches indicate the number of digestive enzymes that are in each of these organisms. So these are the gram-positive firmicutes, many individual genomes from that phylum, likewise for the Bacteroidetes and the proteobacteria. And you can see just by looking at the size of these spheres, the size of these circles, that the Bacteroidetes have a well-endowed ability to digest complex carbohydrates. To put it into a little bit more reference, it's thought that our digestive tract is capable of secreting about 17 glycoside hydrolyses into our proximal small intestine. Some of these organisms have 20 or 25 times that number just in one genome. And if you can imagine hundreds of these species coming together into our gut community, each contributing different suites of enzymes, then it's easy to imagine the amount of metabolic enhancement and plasticity that these organisms can give us. It's also worth pointing out that the types of polysaccharides that flow into the gut environment come from a number of different sources. So we tend to think of diet as a very abundant and dominant source of these. So molecules like starches, plant cell wall, pectins, and hemicellulose, cellulose, all come into our gut from the dietary plant matter that we eat. If you eat a carnivorous diet, you eat different kinds of complex carbohydrates in the form of animal connective tissue. Things like glycosaminoglycans, chondroitin sulfate, heparin. But then again, the microbes themselves are very good at making their own complex carbohydrates, and we don't really know yet what the impact of that biology is, if there are organisms that are beginning to adapt to parasitize the carbohydrates that others make for nutrient sources. The initial answers to that question suggest that yes, they do. But a very important source of nutrients, carbohydrate nutrients that enter the gut, it's cut off a little bit, come from our cells. So if you can imagine 10 to the 14th bacteria existing in high density up here in the lumen of our GI tract, and this is our healthy colonic tissue, we need to protect ourselves from all those bacterial antigens, and we do so in part with the assistance of our immune system, but also by making a physical barrier. And that physical barrier is largely composed of a gel-forming mucus layer that's made up of secreted mucin glycoproteins that themselves have different carbohydrate linkages that are distinct from these other sources. So in terms of just introducing a little bit of carbohydrate chemistry and glycobiology, I always like to contrast the structure of starch, which is up here labeled in red, with the types of structures that are on these secreted mucin glycoproteins. So many of you probably know that starch is a polymer of glucose, so it's just one component sugar, and it's relatively simple in that it has 4 glycosidic linkages and some alpha-1,6 branch points that connect chains of this. So it's simple with respect to linkage, it's relatively simple with respect to sugar, but it's quite large. So from corn, for example, the average starch molecule is somewhere in the order of 1 million of these glucose units. In contrast, if you look at the molecules that make up this gel-forming mucin barrier, they're secreted mucin glycoproteins, and this is a schematic of one shown here from N to C terminus. And the middle regions of these molecules, which are huge, thousands of amino acids, highly repetitive in serine and threonine residues, have very short, relatively short oligosaccharides attached to them that look something like this. So 5 monosaccharides, fucose, galactose, acetylglucosamine, and acetylgalactosamine, and sialic acid. But they can be remarkably variable. In fact, the molecules like this that are secreted into our distal gut, the distal guts of mice, have about 100 different or more structures like this attached to them, which create remarkable chemical diversity for an organism that wants to use mucus as a nutrient source. So the types of questions that really guide my research, going back to my postdoc and are guiding my research now at the University of Michigan, are which microbes of the many that live in the gut degrade these different sources of polysaccharides, what are the mechanisms that enable them to do this, how fine is the level of adaptation to subtle structural variations as we begin to consider much finer groups within each of these categories, how fine is the level of specialization between our diet and our microbiota, do some metabolic behaviors contribute to disease, and I picked these questions, but I'll answer each of them with a short bit of data presentation today. And I really just want to highlight that the long-term goal here is to understand the interaction of this ecosystem with polysaccharide nutrients and other sources that manipulate this, that guide the biology of this community, with the goal of being able to manipulate it. And you can think of diet as a particularly attractive way to intervene with into the biology of the bacteria that are living here, because we can just eat something and it goes right through our distal gut and we can potentially change the behavior. But in order to do that, we first need to understand what that biology is. So going back to the group of organisms that we study, which if you remember on the first slide of the group of gram-negative organisms that are in the Bacteroidetes, the first pieces of data that indicated what members of the Bacteroidetes are capable of, how they're capable of degrading carbohydrates, came from an organism called Bacteroides theta iota omicron. I'll refer to it as B theta for short. This is just one of many Bacteroidetes species that live in our guts, and the samples of humans would suggest that most of us in this room have B theta living in our gastrointestinal tract. It's just one of many species, but it alone is capable of degrading over a dozen different polysaccharides and glycans. So it's got quite a broad ability to digest polysaccharides as nutrients. And this work was done in the University of Illinois in the United States by Abigail Salyers in her lab over the course of most of the decade of the 1990s. And what they characterized was this simple gene cluster right here. So these are 8 genes that they called the starch utilization system for short, or SAS for short. And what this gene cluster encodes is a cell envelope localized system for degrading starch as a nutrient. So the products of these genes are schematized in this illustration up here. The product of this C gene is an energy-dependent outer membrane transporter. Again, these are gram-negative bacteria with an inner and an outer membrane. So this is outside the cell. And this transporter works with 4 other ancillary outer membrane proteins called SUS-D, E, F, and G. I'll show you crystal structures of all 4 of these in a few slides that function to bind that high molecular weight polymer glucose starch to the cell surface. And one of these, the product of SUS-G, is an enzyme that cuts that starch into oligosaccharides that get transported across the outer membrane through this transporter. And then there are 2 additional amylases, enzymes in periplasm that cut those oligosaccharides into pieces. And that's one way that this particular species of bacteria is just one of the many nutrients that it can digest. What's particularly cool and useful about this system is that it's not always expressed. It's only expressed in response to the substrate. So the bacteria economizes its energy, and it makes an informed decision about what it's seeing out here on the cell surface by responding to the oligosaccharide pieces that get transported into the periplasm. And it does that through the product of this sus-R transcription factor that has an intermembrane spanning domain and is transcriptionally activated by maltose. This is a disaccharide of starch that informs the cell that it's not just seeing glucose, but it's seeing smaller or larger polymer glucose that contains some of that alpha-1,4 linkage information that enables it to make the right enzymatic response. And that response is simply schematized down here. So if you look at B theta cells grown on glucose and use an antibody against this orange protein out here, sus-D, you can see that there's very few of these proteins present on the cell surface of these discrete puncta, but if you look just a few minutes later after you dump this culture into medium containing starch, they've massively upregulated the proteins involved in this system and are basically flooding their cell surface, getting ready for during active catalysis of this polysaccharide. So around 2003, Jeff Gordon's lab published the first genome sequence of a Bacteroides genome, and it just happened to be the B theta genome. And what became immediately apparent when the genome sequence of this organism was annotated was that paradigm that I just took you through for degrading starch was replicated over and over and over and over again in the genome of the species. So this is a circular schematic of the B theta genome. These red gene clusters are gene clusters involved in degrading carbohydrates similar to that starch utilization system that I just introduced you to. And this arrow points to those 8 genes that compose the starch system. Many of these other ones are much larger than that. These red gene clusters around this genome encompass most of the 300 or so polysaccharide degrading enzymes that are in this one genome, suggesting that they harness a lot of that enzymatic potential with this transport mechanism to allow this organism to do phenotypically what it does. So just a little bit of nomenclature as I go forward. I'll refer to the protein products of these systems as sus-like systems, and I'll show you in a minute how they're similar to that prototypic starch utilization system. And I'll refer to the gene clusters that encode these red blocks as polysaccharide utilization loci. But you can think of these essentially interchangeably as the genes and the proteins that they encode. And what we now know that we have hundreds of genomes of additional Bacteroides, both from within our GI tract or from the environment, is that there's similar content of these types of systems in other organisms across the entire Bacteroides phylum. So this is a phylum-specific paradigm for complex carbohydrate degradation. So what makes these sus-like systems similar is that each of them tends to have this core transport mechanism. These sus-C-like oligosaccharide transporters and sus-D-like binding proteins are what we use to search genomes for these systems and what we use to annotate all those red gene clusters on the last slide. A pattern that's beginning to emerge is that these systems also have functional homologs, at least, of 2 other proteins. And I'll tell you in detail what these 2 proteins do, but these are functional homologs of sus-E and sus-F, which are carbohydrate binding proteins. Beyond that core molecular architecture, there's a remarkable amount of variability in the gene number, the types of regulators that are associated with these systems, the enzymes they encode. And you can imagine what that variability does is it allows this system to adapt to different carbohydrates. So if you replace the surface amylase with an amylase for a different plant cell wall polysaccharide, correspondingly new periplasmic enzymes, a new sensor regulator that responds to something other than maltose, the system can diverge and adapt to different carbohydrates. And what we now know, and I'll show you a little bit of evidence for this, is that these individual poles do indeed each target their own cognate glycans. Things from all 4 categories, from dietary carbohydrates, from microbial carbohydrates, as well as carbohydrates that are present in our mucus layer. The associated enzyme repertoire is as I insinuated, suited towards the target substrates. And the enzyme number really correlates with substrate complexity. So starch, being that simple polymer of glucose and 2 different linkages, is somewhat easy for the system to break down, which is why it only needs 1 enzyme on the surface and 2 in the periplasm. As the substrates get more complex, you need more enzymes out here, you need more enzymes in here. And I'll show you a couple of examples of that as well. So this slide really summarizes about all of the work that was in my postdoc period in Jeff Gordon's lab. And what I really wanted to do was do an in-depth study of 2 species that have diverse and broad carbohydrate degrading abilities. And those two species were beef theta, here, which I've already introduced you to, and one of its close relatives is also abundant in human GI samples, which is a species called Bacteroides ovatus. And so I picked these organisms because together they degrade nearly everything that we could buy or purify and throw out as nutrient sources, with the notable exception of cellulose. So within these two genomes, we had a broad phenotypic representation of metabolic ability that I wanted to map back to the genomes of these organisms. And what's interesting is that in the genome sequences of these two organisms, out of 198 polysaccharide utilization loci that are contained in the two genomes, only 28 of them are in common. These are fairly closely related strains. There's a lot of uniqueness in each of these genomes, and the uniqueness is shown for beef theta up here in gold around its genome. The names on here aren't important to read. And the uniqueness down here for B. ovatus is shown in blue and in pink around its genome. So it's got a lot of unique genetic potential, at least relative to each other in this pairwise comparison. The key here is that each one of these strains can't do it all. And the pattern that emerged is that I should point out that the experimental evidence here is mostly by gene chip microarrays. So we would grow these organisms in vitro, feed them mucin, feed them a purified fraction of a plant cell wall, feed them starch, etc., and ask which one of these blocks of genes come on. That's all detailed in these two references down here. But for many of these systems we've done follow-up genetics. Both these organisms are now genetically manipulable. We can knock out gene clusters, we can knock out regulators, we can purify proteins and do biochemistry. So we've made a lot of these annotations by that mechanism and are continuing to do so. But the pattern that's emerged, at least in comparing these two strains, is that B theta has many unique gene clusters for degrading the O-glycans that are present in that protective mucus barrier. That's a trait that B. ovatus lacks in its genome. But in reciprocal, B. ovatus has many unique poles for degrading plant cell wall polysaccharides, particularly a class of chemically unrelated molecules called hemicelluloses. That are shown here in pink in this genome, several different gene clusters, and that's a trait that Bacteroides ovatus, that Bacteroides theta omicron lacks. So you can begin to imagine this divergence of these two species into different lifestyles in the gut. Maybe B theta being a good mucus degrader, living down in that protective mucus layer, or at least living off it for a nutrient, and B. ovatus being an organism that's more geared towards living on the plant particles, the dietary material that's flowing through our GI tract. And when I got to the University of Michigan, we wanted to explore this in more detail for sure, beyond just two organisms. So we came up with a system for profiling the carbohydrate utilization abilities of a much broader array of these bacteria. And so this is a little bit cut off, but what we built was a carbohydrate growth array to profile the abilities of these organisms in vitro. And the way this is set up is that there's 47 substrates is the number that's cut off over here. Each of them represented in duplicate and non-adjacent wells with water blanks. We have an anaerobic culturing robot that reads these. And we have 355 isolates here, mostly from human, which is shown in green, and a handful from animals that are shown in pink. And those animals are mice, pigs, cows, and goats. Of Bacteroides species. And so just to orient you, advance one. I'll keep it here. Just to orient you to the type of data you're looking at, each one of these colored bars here is basically two quantitative bacterial growth curves, which are schematized here. And from these growth curves, we can write scripts to measure the exponential doubling time or the total growth rate on complex substrate. And basically, this summarizes about 32,000 individual such growth curves. And at this density of data, you can begin to treat the results like microarray data. You can normalize across what a species does. So a given species behavior is represented as one horizontal line in this heat map. You can then normalize within the substrate. So for these starches, for example, who is the best and who is the worst at degrading that substrate, and begin to homogenize the data that way. And so Cartha Ehrlich was a master's student in my lab who did this analysis once we collected all the data. And the first thing he did was ask, and I should point out that the horizontal axis here, I've organized just by my intuition. So I've asked, as I group these into clusters of similar polysaccharides, so starches and fructans, which are plant storage polysaccharides, glycosamine glycans, plant cell wall polysaccharides, etc. But the first thing he did was ask an unsupervised hierarchical clustering program to take this data for each of these individual 355 isolates and group it together based on similarity profiles. And in tandem with that, we sequenced the 16S of these 355 species and gave them a name based on relation to the known type strains of these different Bacteroides species. And quite remarkably, what happened was that the members of each individual species grouped back together into clades. And one of the nice examples right here is Bacteroides fragilis. Comes in back into a monophyletic clade over here. There's 60 isolates that all group together based on their carbohydrate utilization profile. So they all degrade starch, more or less. They have inulin degradation, which is fructan. They lack most of these traits in the middle, but then they're good mucin degraders. And if you look down at B theta, it kind of expands on that theme. It keeps the mucin, it keeps the starch and fructans, but it adds these new traits in blocks, suggesting that as it's expanding, these organisms are expanding and contracting their metabolic potential. They're targeting similar groups of polysaccharides together. So if you go after the pectins, you tend to go after all the pectins. If you go after the hemicelluloses, you tend to go after all the hemicelluloses. And actually these blocks here are kind of interesting because some of these presence and absence calls actually appear to break down by hemicelluloses that are derived from monocotyledonous and dicotyledonous plants. So the organisms may be getting even a little bit finer resolution than we can even detect just by the way I order these things. But interestingly, there were positive correlations between using carbohydrates from the same source, as I just mentioned. But there's also a negative correlation between utilizing the O-glycans that are present in mucus, and that's present, that's signified by this column over here, and the ability to go after, the propensity to go after carbohydrates that are from a dietary source. And you can prove that with the math that Karthik did over here, but you can see it intuitively just by eye. So as the look down this column of the really good mucin degraders, which stops right about there, you tend to have fewer of these other traits. And as you begin to pick up plant cell wall hemicellulose degradation in particular, this phenotype goes cold and then becomes absent. So it suggests that maybe there's some geographical separation in the abilities of these organisms. If an organism lives in the mucus layer, it may be less prone to adapt itself to live in the lumen of the gut, where these dietary nutrients are a dominant nutrient source. So we really did that study as a hypothesis generating exercise to expand our view of what these organisms are capable of doing. And I want to just go through two examples of the way we use that data to gain some insight into some finer details of these organisms. So the first one is xyloglucan. So xyloglucan is a hemicellulose that's present as one of the major polysaccharides in dicotyledonous plants. So things like solanaceous plants, tomatoes and eggplants, or leafy greens like lettuce. And what's interesting about xyloglucan is that depending on the source that it's derived from, its structure varies. So it always has this beta-1,4 linked glucose backbone, which is similar to cellulose in its architecture. But these glucose units are decorated differently with xylose units that are capped by 2-galactoses in the case of tamarind xyloglucan, or arabinose in the case of tomatoes and eggplant, or fucose in the case of lettuce. And so in looking through just the organisms in that heat map that was on the last slide that have the ability to degrade xyloglucan, this type of xyloglucan, you can see from a phylogenetic perspective, the ability is quite disparate and spread out through this phylum of bacteria. So this is Bacteroides ovatus, which is one organism that we've studied this in, but its neighbors lack this ability, and then it pops up again over here on the tree, and so on and so forth. So not every organism is doing all this metabolism, and it's not just present within a limited taxonomic group. An organism that has it could be surrounded by neighbors that lack that ability. That's shown in a little bit higher detail here. So if you look at individual isolates of Bacteroides ovatus, which are shown here in red, many of them have this ability, but some of the genomes don't have this ability. And if you look at Bacteroides ovatus phylogenetically, its nearest neighbor, Bacteroides xylem solvens, always lacks this ability, at least in the survey of organisms that we have in pure culture. And we've actually gone into these organisms. We have one genome sequence of one of these and many of these, as well as many of these. And you can see the place in the genome where this gene cluster has integrated. And it's integrated, at least in the sequence isolates that we have, at the exact same spot in all of these organisms. So we can use this phenotypic readout to go in and look at the way this trait has evolved within a greater phylogenetic group. Then we can also use it to ask genomic questions to begin to track down these traits within the organisms. And so what we did in collaboration with Harry Brewer's lab in Sweden and at the University of British Columbia was build a more complex model, or a model for how an organism like Bacteroides ovatus would go about the process of degrading this more complex substrate. So unlike starch, now we have a polymer with extra decorations and extra monosaccharides that are appended to it. Those extra decorations require more enzymes for catalysis. So instead of one surface enzyme, now we have two. This one seems to be the most operative. And what this does is it cuts these more decorated polysaccharides in pieces. They get transported into the periplasm. And there, instead of just having two enzymes like you did in the starch system, now there's 6 of them. And what these 6 enzymes do, they're color-coded to the different sugars in this illustration that they would remove. This group removes these aragonoses, and then this GH31 removes xyloses, and so on. And the complex nature of this polysaccharide and the fact that these enzymes work from one end necessitates that they have to work cyclically together. So one enzyme would remove its target linkage and another one would act. So we can begin to build a model of how the organism would go about degrading this more complex carbohydrate. We're targeting now even more complex carbohydrates than xyloglucan to further test this model and ask how the system scales with increased complexity. One of the interesting features that's emerged is that because we have so many genomes of these organisms, so this is the gene cluster in Bacteroides ovatus that encodes that system that I just showed you a schematic for. Now that we found it in one organism and we have genomes of other organisms and phenotypic data, we can go laterally using comparative genomics to ask what are the gene clusters in these other Bacteroides that possess this ability, what do they look like? So we've validated that each of these organisms can grow on xyloglucan, and that each of these gene clusters is expressed in response to xyloglucan. And one of the interesting, very fine level patterns that's emerged is that Bacteroides ovatus lacks the enzymes to go after these fucosylated xyloglucans that are present in the leafy green material that we commonly eat in our diets. But if you start going down this list, suddenly enzymes in Bernie's classification of GH95 and GH29 begin to appear. What the enzymes in these two casein families are alpha fucosylases that would be predicted to remove these linkages. And what Alexandra and Harry are beginning to continuing to test now is whether or not purified forms of these enzymes actually do enable these systems to adapt to a new carbohydrate. You begin to see the genetic potential of how these organisms are fine-tuning their metabolism towards closely related but chemically different polysaccharides. To kind of emphasize that point just a little bit more, I wanted to throw in some data on xylogen degradation. So xylogen is a slightly more complex carbohydrate than xyloglucan, at least from some sources. It's a major monocotyledonous, a major hemicellulose and monocotyledonous plants that we commonly consume, so things like corn, sorghum, rice, wheat. The interesting thing here is that just like xyloglucan, the side chain structures that decorate this polymer, so this is a beta-1,4-link polymer, xylose, with these different side chains coming off it, those structures can vary based on the source of the carbohydrate, and our bacteria seem to know the difference. So this is Bacteroides ovatus again, and if you grow wild-type Bacteroides ovatus on this molecule purified from wheat, sorghum, rice, and corn, it grows well on all 4 of those substrates. It has two gene clusters around its genome for degrading that are responsive towards xylem. One of them has 5 enzymes, another one has 16 enzymes in it. If we just knock out this first polysaccharide degradation locus, we basically retain the sorghum, rice, and corn phenotypes quantitatively with what was present in wild type, but we completely lose the ability to grow on wheat, suggesting that this gene cluster is somehow specialized either enzymatically or at a sensory level to equip the organism to grow on wheat. And we get basically the reciprocal when we knock out the second locus. So remove 16 enzymes from the genome. The sorghum, rice, and corn phenotypes go away. The wheat phenotype is quantitatively diminished, but is still present to a good level, suggesting that there's differences in the bacterial response to different food items that would even related to polysaccharides. So that kind of gives you a bit of a flavor of the specialization of the bacteria to different nutrients. Going back to the prototypic starch system that was the model for these cell envelope associated polysaccharide utilization systems, another line of experimental evidence in the lab is how do these sus-like systems function at a mechanistic level. I'm showing you this cartoon here, which we put together as an initial illustration or model, but we're beginning to really test, particularly with respect to these outer membrane proteins, what the real molecular mechanism of this system is. And I wanted to go through a little bit of that in the next few slides. So, in collaboration with Nicole Porter-Patton, who's also at the University of Michigan, and these structures are all summarized in these 3 references, we've gone about looking at X-ray crystallographic data for all 4 of the outer membrane proteins that are in the starch system. So Sus-D, which you'll remember is one of the hallmark features of these systems, Sus-E, Sus-F, and Sus-G, which is the enzyme in this system. And I apologize that these aren't color-coded with the exception of E and F. Sus-D and Sus-G are different colors up here, so I've labeled them for you. But we've got X-ray crystallographic structures of 4 of these proteins, and the immediate enigma that emerged in this structural analysis was that these 4 proteins that just interact with starch, this relatively homogeneous polymer glucose, have 9 sites spread across them that interact with starch. One of these is catalytic, shown by this yellow and red molecule docked in the catalytic domain of Sus-G. But just adjacent to it is a non-catalytic surface binding site. And then there's 1, 2, 3, 4, 5, 6, 7 more binding sites spread across these proteins. So it raised the question of why would an organism need all these binding pockets to interact with starch. And so Elizabeth Cameron, who worked on some of these structures, was a PhD student in my lab, just graduated last week, wanted to go in and take a more refined approach to investigating that question and leveraging the presence of these crystal structures. So what she first did was go into the Sus-D protein and mutate the residues around this binding site to ask what happens if we specifically eliminate the ability of that protein to bind to its target ligand but keep the protein itself present on the cell surface. And the reason why she asked that question is that we had known that if you just deleted the gene for Sus-D, so took away the protein altogether, the cell is dead on starch. And she did an experiment where she mutated the binding pocket, and that's going to be referred to as a Sus-D star mutation. She got the same phenotype. So the protein was there, but it didn't work, and you got the same phenotype. But basically what she figured out, essentially from contaminated starches that you can buy from companies like Sigma, that if there's a very small amount of saccharide present, this is a controlled experiment where she grew on 0.5 mg per mil maltose, far too little maltose for the cells to grow appreciably on the same level that they grow on starch. You can bypass the phenotype in the Sus-D point mutation, and you get a different response than if you delete the gene. So here the protein is absent, here the protein is present, the binding site just doesn't work. And it suggests, what her quantification of this result suggests was that the growth rate of this Sus-D binding mutant, once it gets going, is essentially indistinguishable statistically from wild type. But it has this extended lag phase suggesting that maybe the function of that single binding site in the homologous SUSD protein that's characteristic of all these systems is important for sensing the available maltose that is bypassing this phenotype. And so in order to address that in a little bit more detail, what Liz did was she made an experiment to measure the transcriptional responsiveness of the B theta cell, either wild type or her SUSD binding site mutant, to different concentrations of starch molecules. So the way she works this is that she grows cells in a condition that doesn't induce the system, which is minimal medium glucose. She washes the glucose away and then dumps them into a condition where there's an inducing oligosaccharide, in this case either multitriose or a heptamer of glucose, multiheptose, at one of these different concentrations. So this is 2.5 milligrams per mil, close to the concentration we grow at, and then several orders of magnitude reduced. And what she does is she measures the expression of the system by quantitative PCR, the message level that's produced 30 minutes after that exposure. And what you can see is that the wild type cell is remarkably sensitive both to the trisaccharide from starch and the heptasaccharide from starch, even more sensitive to limiting amounts of heptasaccharide. But when she removes just that binding pocket by point mutation, the organism becomes several orders of magnitude worse at responding to this substrate, which tells us that its probable role is in seeing these oligosaccharides that are generated at the cell surface by the enzyme and allowing the organism to make a response. When you remove that, the organism becomes a little bit more blind to the presence of these oligosaccharides. And so that was just data for one of these proteins. She asked, what's the role of all the rest of these? And to summarize some data that was in this paper, she knew already that she could eliminate all 5 of the binding sites that were in this SUS-E and SUS-F proteins together and not perturb the growth rate on starch. So we already knew that 5 of these were relatively dispensable. So what she did was she made 2 combined mutants. Both of them were lacking these 2 proteins together. And then on top of that, she tested either a mutant lacking these in tandem with this domain or a mutant lacking these in tandem with this domain. So she'll call this, or I'll call this CBM58, a mutant in EF and CBM58, or a mutant in EF and GSERF to signify that mutation. And what's quite interesting is that on high molecular weight cornstarch, as I pointed out on the last slide, you can lose the 5 sites that are in SUS-E and F and the cell grows normally. You can lose that SUS-G CBM58, the cell grows normally. But when you remove all 6 of those sites, all 3 of those proteins together, then you begin to see a subtle rate defect emerge. It's not great, but it's partial. What was even more striking was that if you remove just the surface site that was on the enzyme on the bacterial surface, this is the SUS-G SERV mutation, the cells slow down a lot. And if you merge that phenotype with the loss of SUS-E and SUS-F, now they're completely dead. So she's removed 6 binding sites and killed the cell's ability to grow on this polymer. And so she asked the same question she did for SUS-D, is this a sensory defect or is it something that happens subsequent to the sensing stage? And so I kept the SUS-D data up here. This was the mutation that lost sensitivity. And in contrast, if she looks at the sensitivity of the response of these 2 triple mutants, on multitriose, both mutants have a slight defect relative to wild type. On a longer polysaccharide like multiheptose, they have no discernible defect. So the defect is not at the level of sensing. It must be something after that, once the catalysis has begun. So she went back and asked what happens on starch. This is now fully high molecular weight starch from corn. SUS-D makes a very poor response to this, as you'd expect from the fact that it's dead on the substrate. But in contrast, these mutants, and this one particularly in dark purple, which is completely dead on the substrate, makes a pretty good response. It's got some statistically significant defects here. But at the highest concentration, what was remarkable was that the transcriptional response by this organism is normal, maybe even a little bit better than wild type. But remember, that's the starch concentration that this organism is completely dead on. So even though it's capable of mounting this transcriptional response, it can't grow subsequently, suggesting that the role of these binding sites doesn't play a role in sensing per se. It augments the ability of the organism to catabolize that substrate once it gets going. And so I think for the last, what time do we end at? 1:30, 1:15?
About 5 minutes.
Okay. I'll tell you one more quick story. Getting at the question of whether or not some of the biology of these organisms could be pathogenic or not. So this is a picture of Dr. Jekyll and Mr. Hyde. We tend to think of, as I introduced very early on, these organisms as being beneficial, being symbiotic, but could they on occasion turn bad for us? And what we initially got into this question, the reason why we initially got into this question was that in the last few years, some stories have been emerging relating to the biochemical complexity and stability of that protective mucus barrier in the gut and what happens if you perturb it. So this is a study in which one of the glycosyltransferases that makes those oligonucleotide structures that decorate protective mucin was eliminated from the mouse genome. And in this case, the mice that are lacking this normal mucin glycosylation begin to get spontaneous colitis, suggesting that some perturbations in the glycosylation structure of mucus can predispose to inflammatory disease. Another paper came out in 2011 from a group at WashU in another genetic mouse model. This was a mouse model called DNKO mice, where IL-10 and T-cell specific TGF-beta receptor signaling were eliminated. So these are two cytokine pathways that dampen the inflammatory immune response. When you eliminate these two pathways, 100% of animals with this genotype are dead by 6 weeks of age due to an acute fulminant ulcerative colitis. You can fully inhibit this disease progression by broad-spectrum antibiotics like metronidazole and ciprofloxacin. And what the investigators found in this particular report was that they could inhibit disease. They determined what they were eliminating were members of the Bacteroides, and they figured out that they could add culture members of Bacteroides back in pure form and reinitiate that disease that would ultimately kill the animals. The best organism at reinitiating that disease was Bacteroides stati omicron, which was interesting because if you remember back to that simple pairwise comparison between B theta and B obatus, B theta was the one that's capable of going after these carbohydrates that are present in mucus. So we did a lot of experiments. We did some experiments actually with the mouse strain of B theta that they originally found, and we weren't able to do good genetics in it. We weren't able to do any genetics in it. So we went to our freezer and pulled out a mutant that I had been working on for a while since my postdoc. And basically what this mutant is a B theta mutant. We call it 6X, BT6X, that has impaired mucus degradation. So to give you the short version of this, we've knocked out 6 consecutive pathways. 5 of them are signified by these ECF sigma notations here. And one is a post-translational activator of sulfatases called anaerobic sulfatase maturating enzyme. But basically what each of these regulators does is controls activation or expression of a subset of enzymes that number 43 in total. And we picked these pathways based on in vivo transcription profiling of this organism in mice as being some of the most active pathways in a mouse gut, and being active towards degrading these different host-derived carbohydrates, either aniline glycans or these oleic glycans that would be on mucus. And so we sent this to WashU and we asked them to put it in their mice. So these are DNKO mice that would get disease that are been treated with antibiotics, taken off the antibiotics, and now these B theta strains are being put back in. This is just a healthy control of PBS, so this is normal healthy colon with good crypt architecture. This is what happens when you put wild-type human B theta in. You get disease, the crypts become elongated, neutrophils infiltrate. But our mutant that we knocked out those 6 pathways in mucin degradation doesn't cause as much disease. Actually, quantitatively, it doesn't cause any different disease from the negative control, suggesting that by eliminating the ability to harvest these mucosal nutrients, we've changed the outcome for the host from disease to no disease. All that's shown quantitatively here. So this is wild type, reduced bars here. This means disease. This is the mutant, no disease. And we broke those 6 pathways up into subsets of mutants, and basically you take everything that was on the left half of that slide, these are the 5 ECF regulators, you still get disease. It's just that anaerobic sulfatase maturating enzyme, a single gene when deleted from the genome, now results in lack of disease and abrogates that ability. We can genetically complement this to restore disease again. So our model is that this enzyme, anaerobic sulfatase maturating enzyme, equips the bacterium to activate its sulfatases. It has 28 of these that we believe go in and remove protective sulfate groups from secreted mucin in the gut. And that's the first step in enabling this organism to get at the underlying glycosidic linkages and cause disease. And that's requires this anaerobic sulfatase maturating enzyme. But when we block this genetically, we shut off this cholinogenic process. And just to quickly end with one piece of data that is gathered from in vitro evidence. This is kind of fun. So this points to the other direction of B theta being an unfriendly guy in the gut. So we've done these simple experiments with feeding B theta pure polysaccharide like polydolacturonic acid. So this is pectin from plant cell wall. And you get this classic single-phase exponential growth profile. If you feed it purified O-glycans, you get the same response. But if you begin to titrate these two together, so 20% O-glycans amidst 80% PGA, begin increasing the O-glycans, you get a very classical bacteriological phenomenon. This is similar to Jacques Monod's lac operant story from back in the 1960s. Diopsic growth, where the organism is eating something first, reorganizing its metabolism, and then eating something second. The second phase here lengthens as you add more O-glycans, suggesting that it's deprioritizing utilization of the mucin over this dietary alternative. And you can test that hypothesis further if you harvest RNA out of this point in growth and this point in growth, and look at the ratio of expression early to late. The genes involved in pectin degradation are on 100-fold more highly early, but the genes involved in mucin degradation are repressed. And so that raises the questions. We've observed this for all the other carbohydrates that B theta eats, with the notable exception of human milk oligosaccharides, which are homologous to, similar in structure to these oligonucleotides. But it raised the question of whether or not other Bacteroidetes have the same or reverse priorities. And recently we found one that has the reverse. This is an organism called Bacteroides bacilliensis, and this organism only grows on starch and the mucinoglycans. Ordinarily, B theta would suppress O-glycan utilization in favor of starch, but this organism suppresses starch utilization in favor of genes that we've determined to be involved in O-glycan degradation. So it has the exact opposite response as B theta. And then we've recently gotten some molecular insight into what drives this mechanism. So this is one pole that has a sulfatase in it involved in O-glycan degradation. And simply by knocking out a very small 190 base pair, in this case, intergenic region, non-translated intergenic region, and we have reason to believe that this is a single transcript that reads all the way through here. We're not removing our promoters, which is what this control is telling us. So wild type and bacteria missing this region both express this in response to oligonucleotides, but ordinarily these genes would be repressed in a condition where we fed the organism fructan, which it prioritizes, until later in growth. But when we remove this region, we flip that response to now where that system is prioritized. So we think the signal to repress these systems is somehow going through this little region of the DNA or RNA over here. We're actively searching for what that mechanism might be. And we think there's more of these spread across all of these Bacteroides genomes. So I just want to end with a couple of summary points and conclusions. Hopefully what I convinced you of today is that these gram-negative Bacteroides that are plentiful in our gut microbiota, more or less universally, as far as we know, universally use this paradigm based on this starch utilization system, these sus-like systems, to go about polysaccharide degradation. But at the individual species level, individual organisms, individual genomes, the repertoire of traits that are present can be remarkably diverse, suggesting that some of these organisms have evolved to occupy distinct niches. At the functional level, specificity lies in multiple areas. So for the enzymes that are present in a system, the sensor regulators that control them, but also the last point that I made, the prioritization of polysaccharides relative to each other, probably all factor into the response of whether or not these organisms are going to deploy their phenotypic and their metabolic potential in the gut. And some of these behaviors can potentially be harmful. So things like mucus degradation, or at least mucus desulfation, may be harmful and pro-collagenic in some cases, at least in that one example I showed you of a genetically susceptible host. And if that turns out to be true, potentially we can use common nodes like anaerobic sulfatase maturating enzyme as drug targets to block the ability of members of the microbiota, even many members of the microbiota, to engage in that kind of behavior and eliminate the impact, their impact on diseases like inflammatory bowel disease. So just to end with a few slides, I think I acknowledge people that did the work as I went through. So these are pictures of almost everybody whose work I showed. Elizabeth Cameron over here did the starch binding X-ray crystallographic work and the mutagenesis. Nick and Karthik are shown down here respectively. They made that heat map. We've had some great collaborators on this project, including on other projects, Bernie and Pedro over the years, and some ongoing works. I'm looking forward to talking more about what to do as we go forward. Looking at these systems, and of course, funding agencies. So thanks for your attention, and I'll be happy to take any questions.
Questions? I do have a question, just while you prepare your question. These polysaccharide utilization loci that you have dissected so nicely there. So somehow there's some cleavage occurring outside the cell. It's importing large oligosaccharides in the periplasmic space. And I was just wondering what happens if you change your xyloglucan, for instance, from the tomato xyloglucan, and you go to the lettuce xyloglucan, all of a sudden you get an extra fucose residue. What if you don't have the fucose hydrolase that is there? What would happen to the oligosaccharides that have been imported? Are they going to accumulate in the periplasm, become toxic, or is there a way out? And would they be taken by another bacterial species?
Yeah, so we can't really address that with any experimental data, but I can speculate. I think a lot of the points that I highlighted with respect to the specificity of these systems could be potentially geared towards preventing those kinds of responses. So in order to be present in the periplasm, fucosylated oligosaccharide would need to first be transported. So maybe the systems that lack fucosidase just don't transport those oligosaccharides.
So you'd have to have a coordinated evolution. If you just lose your fucosidase, then you're screwed.
Which doesn't necessarily agree with what we see. It could be that an oligosaccharide does get transported, but the regulator just doesn't turn the response up. So maybe the fucose blocks the response until an enzyme is there. So in other words, a system could be geared towards seeing a more conserved backbone piece, and therefore it's blind to caps that it can't remove. That might be the most plausible explanation given the data that we have. And in that case, you can imagine if the transporters were promiscuous and just transported whatever curvated structures they could get their hands on and biochemically recognize. And a new enzyme that removes those caps.
Maybe not all enzymes are coated in the pools. Maybe there is some background level of fucosidase from another pool or from something. There has to be a background expression there. Otherwise, it's like the hen and the hen. If you're not able to cleave your oligos to begin with, then nothing would be imported, and you would not be able to turn on your machinery. So I would tend to think that they are. There's a global basal expression of the number of things.
Right. And so we do have that, so I showed that in the micrograph of the glucose-grown B theta and starch-grown B theta. We measured with single molecules that the glucose-grown B theta has literally, it appears to be 10 or 12 enzymes on its surface. So remarkably small number of single proteins at basal level. But when you dump these into medium with starch, the transcription is maximized in 5 minutes. So they have a way of interacting and getting that system up and running very quickly.
Why do you think something was really interesting to me? And I use that as a paradigm of being careful with hypothesis. The fact that Jeffrey Golden hypothesized that the part of the weight gain was caused because of the presence of the bacteria testing and metabolism bacteria species. And in contrast, when you look at the data, in fact, these people get much less bacteria deaths. Do you think it's a consequence? It's because they don't eat much vegetables that you don't get so much bacteria deaths?
I don't think I can address the obesity weight gain story in a short answer, but I'm not one of the people who believes much in that causality. And the reason for that is that the very first slide I showed with the germ-free versus conventionalized animals, the germ-frees are abnormal there. So that's normal, having a normal microbiota, that's baseline. That's 12-ish% body fat. You lose 50% of that when you remove the microbiota. The experiments that they've done with transplant experiments, the microbiota transplants, have been at that germ-free baseline. So you're dropping your starting threshold 50%. And if you look at the magnitude of the responses, they're all coming back up to that normal level, never above. So it's true that in those experiments there's a statistically significant rate difference in terms of the way that adiposity gets laid back down when you suppress it by removing the microbiota. And it's true that those tend to come from obese donors, either mouse or human. But I don't think it's as simple as one microbiota promoting obesity through extra caloric harvest. I think maybe what it could be is that the firmicute-rich microbiotas are just more efficient. Firmicutes tend to have smaller genomes. They tend not to invest as much energy in the gram-negative cell wall that a gram-positive might. That's a bit of speculation.
Yeah, I think that's one of the big mistakes that have been since the very beginning into this story, that we are looking at the flora. We just have the paper ongoing with Donna. We are looking at the flora of the colon, which have nothing to do with obesity. You can remove, as doctors, you can remove the colon and people will not get either obese or stupid or lean. So the problem that the microflora of the colon is just having an effect directly on the mucosa. That's what you say, colitis. Obviously colitis is coming when it's microbiota of the world. But I think my point of view is that anything else that we did based on two stories probably meaningless. Probably just a very small part of it is related to absorption, which is just going in the 51st centimeters of the intestine, not elsewhere. So I think that all the study just based on food probably because they're meaningless.
I tend to agree completely with that. Some of the data would suggest that there's fine-tuning of that response, either through endotoxemia or short-chain fatty acid signaling. But again, I think of it in terms of calories in versus calories out type of equation. Your microbiota is not going to invent, they're not going to make calories where there is none, so you have to, it might change the way we store them.
The thing is that we get much more than, well, definitely, when you look at the size experiments that contain just tens of animals, if you look, there is two big things that I think we need to consider. One is when you look at the farm industry, it manipulates the upper part of the microbiota, there is changing weights. This is very, well, they make so much money out of it everywhere, giving antibiotics in the US and giving probiotics in Europe. So the other thing is that there is no doubt that the fact that you remove all the column and all the bugs, and our machine doesn't change the weight, whatever. So there is, and you know that you can, imagine you know that you can cut the intestine and go, and as long as you keep the 51st centimeter, it's okay, but if you remove this, then you get penetration. So I think that most of the problems that we get, we have not looked at the good target. Of course, the proportion of that come from small intestine to stool is less than 1%. So you're not looking at the good target.
Yeah. Well, probably it's a lot easier to look at what everybody's looking at. You know, the collection of sample is a lot easier at the exit than it is anywhere else.
No, I could see, but I think I can get upper intestine microbial in the stool, but it's coming. I think I can do that. We'll see. Back to it, that's my synthesis is mine, you know that.
Oh, is it?
Of course. My synthesis is the synthesis.
It's the name of Marseille.
It's mine.
You culture?
Oh yeah, of course. We cultured that the first time, of course.
Any questions?
My synthesis is all name.
I have just one very funny question. You are looking at enzyme expressed by the pathogen and you grade polysaccharide from the host. But is there any specificity also toward other pathogen in the intestine? Because there you have like pathogen expressing as well polysaccharide and like conjugates, so they as well affect the surface of the other pathogen.
So
that is antigenic, and why I'm asking this question is because of course the polysaccharide and glycoconjugate are also antigens. So if this pathogen X would bring antigen from the pathogen Y, then you might have a problem downstream. So are they specific to the host, this enzyme?
I don't, so we haven't looked, but I think the permutations that you suggested are all possible. It could be a commensal like B theta you're saying, you could modify glycoproteins on a pathogen. It might even modify glycoproteins on cell receptors that block or enable. I think you can think of it all different kinds of ways. Some could be inhibitory, some could be enhancing, either directly or by modifying host cell receptors. For viruses, the only work that's, some interesting work came out, I guess it was last year, from Justin Sonnenberg's lab at Stanford, showing that B theta, which has sialidase activity to cleave sialic acid residues off of host carbohydrates, doesn't use that sialic acid, so it's incapable of metabolizing sialic acid, but it cuts it off those host carbohydrates to get at the underlying linkages, and it's capable of cross-feeding, providing that free sialic acid to pathogens like Salmonella and Clostridium difficile to enhance their colonization. And they did some elegant genetics in the Salmonella to show that simple pathway, just providing a high-value nutrient, could augment pathogenesis. So,
yeah, because why I am asking that as well is because, for example, I'm thinking about FPS or this kind of glycoconjugate, and mostly glycolipid, and the hosts don't have the enzyme to cut this glycolipid. And somehow now we know that glycolipid are antigenic, so I was wondering whether actually we have found the bacteria could process this glycolipid to
I think it's certainly a possibility. We've done some feeding bacteria, these LPS, and haven't found them with frozen. But it depends, of course, where you get the LPS from.
And the last question is whether, I mean, these things like we, of course, induce, what you are showing that it can induce maximum inflammation as well. So can we, I'm very naive on that, but can we imagine to, for example, cure the gluten intolerance patient with adding some bacteria that will just cut this molecule? I mean, you are showing that it could be.
Yeah, I think you have to bypass the linearity of the gut issue. So if the response to the origin response is in the proximal gut where microbial density is much lower, then you might still have a problem. I don't know too much about celiac and wheat intolerance.
Okay.
You cannot say to bacteria what they have to stay. They do what they want. So you can give that to bugs, but they do what they want. They want to make.
Sure, but they have co-evolved with us. It's not just anything that is there. So, you know, you were saying, you know, that the role of the colonic bacteria is merely to digest what we know. Sure, but in addition, they have been co-evolving with us. We have learned how to live with them. They've learned how to live with us, and this is perhaps what is at the origin of this population, just to recognize somehow, or just to push out some pathogenic bacteria that do not belong.
Not only this, we just have an ongoing work on this. I was really surprised. I can't believe it was so easy. We look at the people before and after coloscopy, and then we completely clean out all the content. There is no more bugs, very, very few. And we look at the composition before and let's say 24 hours after the coloscopy, where they get stool that composition exactly the same.
Exactly the same.
Yeah.
So we think we clean.
So we kill, 10 to the 15 bugs. We come back exactly the same. That's
quite
there is an equilibrium. That's
It's similar to what was seen with the hand washing. There's been some experiments a few years ago where you wash your hands thoroughly and just you wait for half an hour and they're back. It's just amazingly resilient. It takes very little. Well, I want to thank everyone and thank Éric once again for coming to us and for an excellent seminar.